Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Hello everyone, welcome to my YouTube channel. So today we are going to discuss about cell numbers and growth rate. Now we know that how the cells can be used for therapeutic agents. Now before we to use the cells for a therapeutic agent, we must know that whether the required number of cells are present and whether the growth rate is matching in order to get the desired tissue. So for that we should be knowing how many number of cells are called as the range, the normal range of the cells which a particular tissues or organs has got. So once we know those numbers, then we can plan accordingly how many cells we need to, in order to achieve those numbers, how many days we need to culture it. All those things need to be planned in advance. So for that we need to understand the particular human tissues, organs well, so that we can see what are the range of the cells in a particular human organ, and then we need to go for the tissue engineering. So for that this particular topic has been introduced in your syllabus, the cell number and growth rate. Now the cell density, let's start. So the cell density in the human tissue is in order of 1 to 3 billion cells per ml. This means at per ml we are having 1 to 3 billion cells. Can you imagine, with that so much small space we are having almost 1 to 3 billion cells. Hence human body consists of around 100 trillion cells. Can you just imagine 100 trillion cells? But if you consider an organ, a particular organ, it consists of few hundred million functional subunits. A particular organ, for example, take for example liver. So liver will obviously will consist of some million functional subunits. Same thing goes for the brain, same thing goes for the kidney. Okay, so number of the cells is determined by the functional ability of each subunit. How, for example, if you are talking about the kidney, we will the functional subunits of kidney is what? Nephrons. So we will be taking the count of number of nephrons in the kidney. Okay, so here functional ability is what? In order to purify the blood. Okay, wait a minute. Okay, but if you consider an organ, the number of the cells is determined by the functional ability of each subunit. Now each subunit in kidney is what? Nephron. Each nephron's function is to purify the blood, take out the toxicants or waste which is in the blood, and then that can be used. So for example, in kidney the number of nephrons, that is functional subunits, is determined by the maximal clearance of the toxic products. That means the function of the nephron is what? Clearance of the toxic byproducts, and also the clearance of ability of each nephron. So that is how we are going to see. If the filtration is proper, then the number, the actual number, whatever the range is there, that number the kidney has got. If the filtration is not proper, then there are lesser number of nephrons. It's an indirect approach. So for cell based therapies, certain number of cells is required, and it varies depending on the type of tissue that is being affected. Okay, these are some of the examples. For example, chondrocytes. When we are talking about cell based therapies, the example for the cell based therapies, chondrocyte transplants that require few tens of millions of cells. Okay, and another example, lymphocyte therapies, that requires 500 million of cells. For liver support, it requires 10 billion hepatocytes, and bone marrow transplantation requires a few billion cells. These all things are what are, the maximum cells required to do the minimum function of the particular organ. Okay. Now there are certain limitations in the production of the primary cells. For example, a single cell can produce 1010 to the 1015 cells in a culture. However, this depends on the type of the cells, whatever cells you are going to use for a differentiating, because fibroblasts and skeletal myocytes, they grow well in culture, while hepatocytes and beta islets still they do not grow well in culture. So usually primary human cells can undergo 30 to 50 doublings, that means 30 to 50 times they will double or they will differentiate in a culture based on the age of the donor. It obviously this particular in the culture 30 to 50 doublings, that is the range. Obviously this 30 to 50 that depends on the age of the donor. If the more age, then the doubling will be less. If the person is very young, 50 doublings we can expect. If he is intermediate age around 30 to 40, then we can expect 40 doublings. If the person is going beyond 60 to 80, then we can have the 30 doublings, like that. Now this particular concept, that doubling is determined by the Hayflick limit. Hayflick limit is nothing but what? The maximum number of times the cell undergoes for a particular differentiation or doublings is called as Hayflick limit. That will be the maximum limit. Later it will not differentiate. So primary cells vary in growth rate. For example, the doubling time of hepatocytic cells are 11 to 12 hours, while dermal fibroblast requires 15 hours. That means you see the hematopoietic cells is taking, for doubling it is taking around 11 hours, whereas the same different cells will take a different time. That means, for example, the dermal fibroblast for skin cells, they will take 15 hours. It depends on cell to cell. So all these things need to be understood properly, and then you have to conduct the particular set of experiments, and then the optimized parameters, all those things should be taken care, and you get the particular product what you want, and then you use it. So adult chondrocytes, they require around 24 to 48 hours for doubling, while hepatocytes do not grow in culture at all. Okay, so these are some of the things we need to keep it in mind, so that the particular, first we discussed about the particular numbers of the cells, then we discussed about the maximum limit, then we discussed about the Hayflick limit, that is doubling, 30 to 50 doubling based on the age. Then it is about the hours, the time it is taking for each doubling. Then we are having the cells are cryopreserved. After once you get the cells, you can preserve them. You cannot use directly, no. So for example, if the patient has come, we cannot ask him to stay until I clone the cells, then I do the procedures, then I get the particular things done. So it takes a lot of time. That's why even in autoimmune, what to say, the auto cells are that is the problem. Autologous cells, wherever we are using the same cells from the same for a same patient, it takes a lot of time, because a lot of procedures need to be followed, a lot of cells need to be grown, so it will take a lot of time. So that is one of the major drawback of the autologous cell transplants or autologous transplantation. So cells need to be cryopreserved. That's why what happens in advance, if for a major diseases and all, they will ask the people voluntarily give their bone marrow, their cells, so that will be cultured and kept aside, cryopreserved. So that, because these are the some of the cells where a lot of people require regularly. For example, in India is called as a diabetic country actually. So we need a lot of pancreatic cells. So we already know that. So we will most of the companies, or most of the pharmaceutical companies, or even most of the research and development industries, they can collect the cells in advance of different persons voluntarily, so they can keep it aside. So if anything is family person, or if something is happening with the same issues and all, the same family members also cells are also already present. So those cells can be used. Or if it is not the same person or the same family, then the other person which is closely related or something like that, that can be used. So cells are, for that we need to store them. So storing, we cannot directly store normal refrigerator. So we have to use the cryopreservation. You know what is cryopreservation? It is like almost taking back into the very negative temperature. Okay, minus 240, minus 50, minus 30, minus 40, depending on the cells and its characteristics, we will preserve it. So cells are preserved, cryopreserved with suitable protectants. We need to not only just for freezing them, but there should be a cryo protectants also, even at a lower temperature. Also, that should not affect any there should not be any much effects on the cells, like glycerol and dimethyl sulfoxide can be used as a cryo protectants. But however, the solution effects have to be taken care, okay, to avoid the intracellular ice formation during the freezing. During the freezing from cell to cell, in intracellular this thing, there should not be any ice formation. That should be taken care. But viability of the cells after cryopreservation process may vary 40 to 95% of the 95%. Can you imagine? That means, imagine I am keeping the cell today for cryopreservation. After 1 year I am using it, then its efficiency can its functionality can reduce till 60%, or 60% cells can be almost considered as non-functional or a dead cells. Only 40% I can get. So this range comes from 40 to 95. So sometimes you get 95% healthy cells or viable cells or functional cells, whereas 5% are dead. So the range is very very large here, broad here, 40 to 95%. So for treatment, the cells are usually injected into the portal vein, that portal vein of the liver, or it can be encapsulated in a suitable polymeric scaffold and then delivered into the patient. So this has to be done. So these are the two different ways of delivering. Either you can directly inject into the portal vein, or you can encapsulate. That means you use some polymer scaffold so for the encapsulation or the protection, and then you deliver back into the patient, like lipids or polymeric substitutes or carriers. So that was about the cell number and growth rate. Okay, so those are the different numbers, growth rate, how exactly it affects, like that the growth rate in the sense at different temperature, different time, 10 to 12 hours, whatever we discussed, all those things, and cell viability, cryopreservation, everything. So next topic we are having measurement of the cell characteristic, that we will discuss in the next class.